Good day to all the viewers. Today we are going to learn about cytokines. Cytokines are low molecular weight signaling molecules produced by our immune cells. They are regulatory proteins which are glycoprotein in nature. Their main function is to assist in regulation and development of immune effector cells in our immune system. Let us see how they work. Upon inducing stimulus such as inflammation, the cytokine producing cell produces cytokine. The produced cytokine binds to the specific cytokine receptor of the target cell. Upon binding, it induces a corresponding signal transduction pathway and leads to gene activation. As a result of gene activation in the target cell, various biological effects occur. Cytokines act in three different modes. When the cytokine produced by the cell acts on the same cell, it is called as autocrine action. When the cytokine produced by the cell acts on the nearby cell, it is called as paracrine action. When the cytokines produced by the cell enters the circulation and acts at a distant cell, it is called as endocrine action. With the three modes of action, cytokines exhibits different mechanism of action. They are pleiotrophy, redundancy, synergy, antagonism, and cascade induction. Let us discuss in detail. When a cytokine produced differs in the effect based on the target cell, it is called pleiotrophy. For example, IL-4 produced by activated T helper cells leads to proliferation, activation and differentiation of B cell. Whereas when IL-4 binds on thymocytes and mast cells, they lead to proliferation. If we see the cytokine interleukin-4, it has different role based on the target cell. When different cytokines produced by the same cell leads to similar effect, it is called as redundancy. For example, IL-2, IL-4 and IL-5 produced by activated T helper cell leads to proliferation of B cell. Here, in this case, one biological effect is mediated by more than one cytokine molecule. In case of synergy, the effect of one cytokine is enhanced by other cytokine. For example, IL-4 induces class switching of B cells for IgE production. This is enhanced by the presence of IL-5. When the effect of one cytokine is inhibited by another cytokine, it is called antagonism. For example, IL-4 produced by activated T helper cells induce class switching to IgE. It is inhibited by interferon gamma. Cytokines produced by one cell act on the other cell and leads to produce different type of cytokine. And the cytokine produced by the target cell act on the target cell too. This is called cascading effect. Interferon gamma produced by T helper cells activate macrophage to produce IL-2. The produced IL-12 induce activated T helper cells to produce interferon gamma, TNF alpha, interleukin-12, interleukin-2 and other cytokines. These cytokines have different clinical applications. Interferons are used in treatment of viral diseases. Interleukins are used to enhance immune function in patients with immune dysfunction. Interleukin-2 is used for treatment of cancer. Apart from this, various genetically engineered cytokines are used on various diseases such as rheumatoid arthritis, hepatitis, leukemia, sarcoma, and osteopetrosis, etc. It is also used for production of myeloid cells after the bone marrow transplantation for stimulating platelet and RBC production. Thank you.